sequencing in around 2012. That was few projects, but the most important project was UNC-Seq. This slide is from April this year, so we already have enrolled more than 1,000 patients. We completed about 436. Probably right now it's more than 500. However, the most important thing that we learned a lot during these 2 years. So this is the timetable for this type of projects, and as you see, the first phase of the project that between the patient consent and return result was very long. Later, last year we have 56 days, and right now we have around 21 days. But still, this is a lot of time. So we learned that we need to change our approach, and right now, for example, in UNC Hospital, we are doing panels using Amplicon strategy, not a DNA capture like here, and patients which will get not recognized mutations going to the trials for the UNC-Seq, and then we have panel around 2,200 genes which we can use and collect information about mutations. But also very important is that we want to move forward, and there are a few questions which we have. For example, after treatment, how we can monitor progress of the treatment, how to monitor patients after treatment, and very important for me is how we can use screening for population and detect early stages of the cancer. So, of course, the answer for that is liquid biopsies. What are liquid biopsies? Probably you know. We're collecting blood, we are separating plasma, we are extracting DNA from plasma, and we are analyzing DNA content. So people knew about circulating DNA a long time ago. So in even 57, people detected DNA in lupus patients. And in 66, was defined that indeed this is DNA circulating, but they didn't detect that in normal humans also DNA circulates. And it's in 75, new methods for detection, more sensitive, were used, and Steinman was able to detect DNA in plasma in 75. So right now you can find about more than 1,000 papers about different approaches and how to collect, how to analyze circulating DNA. So what is the source of circulating DNA? And this is really a little bit gray area. There are different opinions, but we know that we can detect in oncoexosomes, we can detect probably DNA from necrotic and or apoptotic cells. The most important is that the DNA is fragmented, and for example, this is alternate mechanism how that can happen. But I don't want to go inside of this because that will take a lot of time. However, the most important is that DNA is fragmented, and that has huge consequences. So let's imagine that we have plasma, yes, and we have cells in plasma and exosomes. So we have DNA, microRNA, proteins. So those are our markers which we can use. Also, we can isolate exosomes, which is not easy thing to do, but doable. But the problem is that we have also cells. And if cells start bursting and releasing DNA, we have huge contamination. So first problems always was to collect and how to keep intact the cells and separate them from the plasma. So according to our calculation in normal humans, we have around DNA from around 714 million cells. This is which corresponds 50-100 micrograms of DNA circulating all the times, and it's exchanged every 2-4 hours. So this is a lot. Why we are interested in analysis of the DNA in plasma? so this is like dream, yes, what we want to do in the future, and that triggers our methods which we want to use. So it's really, we think that in the future we will be able to collect plasma from patients which are not diagnosed. We will be able to perform sequencing, and we can give physicians information where to look for potential cancer. As you see, many cancer has a certain correlation with mutation in some genes. Some of the cancer does not have correlation at all. But in most of them, we will be able to find correct markers and try to help physicians to find real problems. Also, I'm not going to talk about parental testing. It's a little bit, that will be talked probably later on. We also trying to use circulating DNA in plasma for other applications, like for example for detection of DNA from transplant, and to figure out when rejection can happen and what conditions, and that can be a marker in future for this type of diagnosis. So there's a lot of assays right now which can be used for analysis of free circulating DNA in plasma. But all of these assays have one common thing. They are based on PCR, which is great because PCR is very sensitive technology. However, it's very limited technology because you can really analyze certain mutations, not whole exomes. To analyze whole exomes, you need to use next generation sequencing, but sensitivity of next generation sequencing is limited. So what we decided to do, we decided to improve sensitivity of next generation sequencing. Of course, this is our calculation because we need to have some certain baseline. What is important? So it's important is that when we are collecting plasma, we are assuming that we are collecting 1 milliliter of plasma. In regular humans, we can get around 10 nanograms of DNA, which corresponds to 3,000 haploid genomes. Okay? So we know also that cancer patients have more DNA, or other patients with other diseases have more DNA. However, this is our baseline. So what we are really analyzing, we want to detect all 3,000 molecules. So, and this is 3,000 molecules, let's say, if you are interested in few mutations, it's easy, but you can imagine that we are investigating whole exomes. So this is a little bit challenging. So we decided, okay, we will do around 10,000, 15,000 X coverage. We're still working with statisticians to determine what will be the best coverage for us. Because time is flying, I will move a little bit faster. We constructed libraries from using TruPlex kit. This is from Rubicon Genomics. And during construction of these libraries, we didn't size select, and also we did not sonicated DNA. So this is what is flowing in what we got from patients. And this is, as you see, those are really fragmented DNA, but this fragmentation is not random. So we think that, and many people also think that what is flowing in our veins is really DNA, which is protected by histones, which is okay, but it also gives us some limitations, and I will talk a little bit later about these limitations. So what we saw after alignment to the reference genome, of course, that we have most of our DNA inserts is around 150 bases. There are some fraction a little bit higher which correspond to nucleosomes which are close together and protecting larger fragments around 300 bases. And what we saw that when we take, for example, 2 nanograms of DNA, we will construct libraries and we are capture, we are able to do pretty good job with the capture. What worries me always that if you are capturing from the very small amount of DNA, you have a lot of duplicate reads, yes? But right now we have something which each lab needs to determine, okay, we are keeping duplicate reads because we know that DNA is fragmented and is surrounded by nucleosomes, and we don't want to lose this information, or we are just to remove all duplicate reads and we will analyze pure data. And different labs approach this thing in different ways. So we decided to deal with this a little bit different way. And also this is another thing which I need to mention. So here down is a whole genome sequencing from the same sample. What you see here that even if this is very light sequencing, if there is a niche like Valley that there is not a lot of reads, we can still using capture technology, we can capture enough DNA to make a calls. Of course, you need to think, we need to have a coverage 10, 15,000x, so that will be very, very challenging. But what I want to point here is that what if we'll design PCR assay instead of exome capture? So we need to design primers, and that will be really difficult in this case, because if we'll design one primer here and second primer here, you see that there is not a lot of reads which represents really in this area. So how we can deal with it? We really right now don't know. We never compare exome capture data to Amplicon data because of the one thing. We don't have right now technology to count real molecules, input molecules. And also what you see here that sequencing has this sensitivity of the sequencing depends on how accurate reads are. And of course, if you are analyzing, for example, repetitive elements, you will see that you have a lot of random errors. Many times they are corresponds to low, 5 minutes, okay. They correspond low quality reads, but in many cases they are very high quality reads. What we decided to do, we decided to system errors. Okay, so PCR also bringing two things: PCR bias and PCR errors. And of course, PCR bias that, for example, from this molecule you will get this number of copies, and PCR errors that between these copies you will find just natural errors. So what we decided to do, we decided to deal with all those problems, and all those problems can be approached a little bit different. I will show this slide later. Using molecular tags. What are molecular tags? In many companies they're calling molecular unique identifiers. So these molecular tags are random sequences, and molecular tagging is tag individual DNA templates with random oligo before PCR or sequencing. So you are adding a random sequence to each molecules, and you are amplifying this. So this is, for example, molecular tag, yes? So for this, we're collecting all molecules with the same molecular tag, and we are aligning these molecules, and as you see, G. So that means that was error during the PCR. So we are collapsing this, and we are getting consensus. This way, we are improving sequencing. Also, there was a huge problem how we can deal with the how many nucleotides we need to use. So let's say when you use 13, we can deal with this. We can deal with 25,000 molecules without major problems. Of course, you can ask 13 molecules is like 67 million possibilities, but this is not true. We are counting how many times we can find in one place the same two molecules. We are, of course, created also software which can deal with this. And this software, how it's dealing when we're doing PCR, and this is mostly for Amplicon methodology, we are overlapping sequencing. So we are creating one sequence first, collapsing these two sequences. So that improves by itself quality, and this is based on the quality score, of course. And after that, we are categorizing molecular tags and also improving sequence, and we are getting final consensus, and we are counting this consensus. And for example, when we use this strategy for metagenomics data, what appeared, this is old strategy which we used always, and this is new strategy. And first thing that we are detecting less different molecules using our strategy. And second, if you will use plasmid with single copy, using strategy because of the PCR errors, we are getting a lot of possibilities, a lot of OITUs, taxonomical units. Here we are getting only one. So we really improved our quality of our data. And this is the last, I need 2 minutes, so 1.5. So for exome capture, what is our, how we deal with exome capture? So really the bottlenecks in all technologies which are using sequencing is library preparation, yes? So we are calculating that around 40% molecules are really, we are using for investigation. Because rest are not well done libraries with molecules, with not ligated adapters. And especially for if you're using 10 nanograms or less. And as you see here, this is standard protocol. So when you're doing PCR, like many cycles of PCR to reach 10, 15x, you are calculating everything, and you cannot calculate how many genomes you used, yes? If you are using strategies with molecular tags, for example, if you from 3,000 you generated 1,300 genomes, you are still collapsing everything to real number of the genomes, and you can count how many genomes you are analyzing. And of course, for that, we had to design special primers. And that was not really easy, but there already one is described in literature. This is duplex MT adapters. We are design our own, and I have to say that it performs a little much better. The reason was that this first one was it's very difficult construction. Ours are very simple, and as you see, we have really efficiently created the libraries for sequencing and for analysis. And I just want to stop it here. We don't have, right now we are doing final capture of molecular tags, and we'll get data probably in 2 weeks. Thank you very much.
Thank you. We have time for a few questions before break.